Right. Welcome back, everybody. Hope you're doing well. So, today we're going to talk about this package called BioPython, which is basically a bunch of tools that help you deal with biological data. Okay, it's really a big package. I'm going to try to give you the highlights, and the rest is for you to explore. Before that, though, I wanted to do a little warm-up with object-oriented programming a little bit, just so that we're all on the same page. And I've been seeing some common mistakes happening with the assignment so far, so we'll do a little exercise, okay? The exercise is very simple. It says to write a class that represents a student. Okay, so I always keep using this example of keeping track of students. So you have write a class called student that has two attributes, okay? A name attribute and a grade attribute. Right, and another class is going to have the name course. It's going to be called course, and it's going to represent it's going to be a collection of students. Okay, so it's going to represent all the students in a particular course. All right, so at McGill you can have different courses, and each one of those courses is associated to a bunch of different students. Okay, and each course is going to have, for example, the attribute course name. So the name of the course we're going to want to know, and obviously a list of all the students in that course. And actually, let me add something too, a max capacity. So a maximum number of students. But this is actually going to be a class attribute, okay? Just so we start to distinguish between things. All right. So this course class that we want to write is going to have instance attributes, course name, and a list of students. So every course is going to have its own name and its own list of students. But there's going to be a class attribute, which is going to be a max capacity. Okay, so every class then is going to have some fixed capacity, which is the maximum number of students that can register in that course. Okay, so we're going to look at the difference between an instance and a class. All right, and the course will also be able to compute the average of all the students in its class. Okay, so it's grade, the average of all the grades of the students. All right, so if we wanted to code this up, this is sort of what that would look like. We're going to be using statistics because we're going to compute means, but the rest is pretty straightforward. So the first thing you do is you define the simplest, kind of most fundamental object here, which is the student. All right. From the student, we can build collections of students, but first we need to have that first indivisible object. Okay, so we have a class, right? We started off with our keyword class, and we give it a name. And like I said, we could just leave this blank, and we would be able to create objects of the type student, all right? But obviously that wouldn't be very interesting. We want to keep track of these things, the name and the grade of each student. So we have to define our own initialization function, right? Which gets called every time you create an object from this class, so an instance. This function gets called, and it accepts an object of this class, so it gets made for you. Before this initialization function is called, actually Python creates an object, all right? And you get to use it as input to your function. We give it the name self, but you can give it any other name. And so when we initialize a student, we need to know the student's name and the student's grade. Okay, so when a student's born, that's what we need to give. Okay, and all it does is it sets some attributes of every instance of this class. So for example, whichever object this function is being called on, aka self, is going to have an attribute .name. And we're going to set that equal to whatever is passed to us when we initialize the object, okay? Same thing for the grade. The student's going to have a grade attribute, and this gets passed to us when we call the initialization function, all right? So now if we wanted, we could create a student, all right? So let's say, let me comment this out for now. All right, so you can say, student equals some. When you initialize an object, you use the class name and you give it whatever the initialization function requires. It requires three things: self, name, and grade. All right. The self object gets given to you for free by Python. Whenever you call whenever you create an object, it gets made and it gets sent to the init function. So you don't have to worry about that. Python creates the object for you. All you're doing here is sending some attributes of the object. Okay. So it needs to have a name and a grade. So let's just give it that information. So let's say it's Carlos, and the grade is 3.4. Okay? So now we should have, if I ran that correctly, okay, no errors. Python probably stored that object and it gave it the name stud. Okay, and then we can say print that object. Okay, and we get this funny looking string, right? Which is basically, Python doesn't know how to print this out nicely for you. It knows how to print lists nicely and dictionaries because those are very standard things. But when you create your own object, all it says is, look, I have an object of class student, and this is kind of its ID, okay? Now, what I'm seeing from a lot of people in the assignment is that, they're doing things directly on this object. For example, the population class that had an attribute which was a list of sequences. People were saying, like, for example, for RNA in self or something, right? Right, this wouldn't work because, this is just an object, okay? Just like if I wanted to, I don't know, it's basically like trying to do a list over something like this, okay? When you really want to get some information out of an object, you usually have to access one of its attributes, right? So if I wanted to know the student's name, I would say print.name, okay? And it would give me back the name that I used to initialize the object. We'll see more about that when we do this next class, okay? So now we've defined an object type, and we can create as many of these instances as we want, right? So this creates one instance, right? I could make another one if I wanted, right? I've made another instance of that class, right? Now, so that's great. We have students. We can keep track of their names and their grades. Now we want to put students inside of a course, okay? Well, it's just going to be another class. It's another type of object, course, all right? That has some attributes that define it. So we make our initialization method, right? And here, all we need to know for the course is its name, all right? And also, we had a max capacity, okay? Let's say max 3 students per course, okay? All right. So this says this class is going to have an attribute max capacity, okay? And it's going to be 3. So this variable is shared by every instance of the class course, okay? Now the instances, however, the init, right, they're going to have their own values for the attributes we define inside the initialization function, all right? So in particular, whatever object that belongs to the class course, in this case self, its attribute course name is going to be set to name, whatever name we give this course, and it's going to have a list of students in it, right? And when we start off the object, it's just going to be an empty list, okay? Is that clear? All right. So that's just how we generated a new instance of the course, right? So we can now have different types of courses. We can have Comp 364 and, I don't know, Math 200 or whatever. So now we can keep track of different courses, and inside those courses, we can put students, right? So we're going to want to actually do that. So we want a method here called addStudent, okay? Let's say we have this method. And addStudent, because it's taking this self argument, it's going to be an instance class, instance method. So that means it acts on instances of the course class. So every particular course, like Comp 364, is going to be able to call this method, addStudent, and do some stuff to it, okay? So this method takes as input a name. This would be a student name, right? Let's just be clear. The name of some student, and that student's grade, all right? All this function does is say, all right, well, we have this attribute self.students, right? Because when the course object was created, we set it to be an empty list, okay? So we assume that this already happened, and that we have some list that corresponds to this attribute students, okay? And we want to modify this list, so it's a mutable thing, right? Lists are mutable, so we can just append something to it. So we're just going to append an object, right? Lists are just collections of objects, and you can append objects that Python creates for you, or you can append your own custom objects. So here we're going to append our own custom object. In this case, we're going to create a student with a name and a grade, and append it to this list of students, all right? So now this particular instance of course is going to have one more student in its attribute students, okay? We also want to, for example, compute, have a convenient function to give us what's the class average at a given point, all right? And again, this is going to be really just taking every student's grade and averaging it out, okay? Since we have this list, self.students, that has all our student objects, and each of these student objects has an attribute grade, we can access every student's grade, right? So all I did here is I'm creating a list, right? Where for every S in the list of students, self.students, I obtain its grade, okay? So this is now a list of grades from the current object's list of students, all right? And then once I have that, I can just give it to the module statistics and use its mean method, and it would return the average grade of the class, right? Oh, but right now we haven't used this max capacity thing. So, for example, let's say we wanted to add a student to the course and it reaches the cap. McGill courses can't handle more than 3 students. So we shouldn't let whoever calls this function to actually add a student, if that's the case. So we can say, all right, if self.students, right, the length of that, so this will tell us how many students there are in this current course, right, plus 1. So like, let's say if we were to add this student, it's greater than course, right? So now we're accessing an attribute of the class. So we give the class name, course, dot max capacity, right? This means if we were to add one more student to our list of students, and if that would go over the class attribute max capacity, so this is going to be the same value for every instance of the class, then we say, you know, class is full, right? And then maybe we just like kill the function. So, oh no, it's not kill it, just say else we do this, okay? All right. So let's play around with it and
Sure.
yeah.
Just to name, change in the estimate, change the estimate underscore name.
Here?
And below your else.
Here? Yes, you're right. Thank you.
But
Yeah. Right, so I'm taking the student's name and trying to add it, so they need to match up. Good catch. All right, so let's create a course. All right, course is Comp364. All I need to create a course is a name, right? And then this function gets called with a particular name that I give it. In this case, it's Comp364. And we're going to start off an empty list in its attribute of students, right? So we can, like, you know, print, for example, c.name, print c.students, right? Course name. All right, I changed it. Course name. Okay. So we printed out that the course is called Comp264, and currently it has no students, right? So it has an empty list there. Now, what's the difference, for example, if I said this, course dot students, right? Would this work? Right? What's the difference between saying course.students and c.students? Yeah. So course is the class, the type, right? And it can also have attributes, but right now the only attribute we define for it is this one, max capacity, okay? It doesn't have an attribute students. Only instances of it have at that attribute students, okay? So hopefully I'll get an error if I do this. Yeah. Right, so course has no attribute students. All right, so this is the difference between a class and an instance, okay? We have one class, right? So it's kind of like the abstract category that it's in, right? That can have attributes. For example, you can print the max capacity, right? That would work. It would tell us that there's a maximum capacity of 3, right? But individual courses that come out of this class, they're going to have the attribute course name and students, okay? So be careful in the assignment, right? When you're doing things like, I think I made a mistake in the description of one of the functions I had to write, where I told you to do something like RNA.replicate, right? Replicate is a method that acts on individual instances of the class RNA. So that wouldn't work. It would be like, I have to make some new RNA, right? Like this, and then this RNA would be able to replicate, right? Okay, so just careful distinguishing between a class and an attribute. A class and an instance, all right? And now I can call, right, I can say this course, right, it has a method add student, and I can add, for example, Carlos and Chris to it, right? And then I can print the average of the class, right? And it says, okay, this class has 3.7 average. Now I can make another instance of the class course. For example, I'm going to call it B, right? And I'll call it, I don't know, comp 202, right? And I can say b.add student. Let's say I'm also in that course, and I'm doing a little worse there. I got a 1.2. Right? So now these would be two separate occurrences of the class course, right? And now they should have different looking lists of students. So if I were to print b.students and print c.students, I should see different things, right? Yeah, so I'm printing kind of the student objects, right? But if I wanted to get a better look at what that was, I would probably have to make a list and say, all right, print b.name for b in, student.name for student in b.students. So I want to get the attribute name of every student in the list of students for the first course, right? And I can do that, I can do that for the second course, which is called C, whatever the first course is called C, right? And I should say, okay, Carlos is in comp 202 and in comp 204, comp 364, and so is Chris in comp 364, okay? So instances are not the same as classes. And that was basically the main point here. Any questions there? All right. So let's jump into BioPython. So we have this really useful library. I guess it's a package written in Python that helps with a whole bunch of different things that biologists might be interested in doing. Okay. How many biology, biochemistry, bio-something students do we have here? Yeah, quite a few. Okay. All right. So most of you will find this fun. If not, don't worry about it too much. It's just really showing you cool things. All right. So it's a third-party package, so it's not built. It's not in a standard library, so you'd have to install it. It's just a bunch of people that wrote some Python code and put it on a website. Anaconda lets you install this from the command line or the terminal, so you can say conda install BioPython. On a Mac, you just open your terminal and you say conda install BioPython. Okay. On Windows, similar thing. It's just called the command line as well. Yeah. Anyway, same thing in Windows. As long as you've done that whole setting up that I showed you guys a few classes ago. All right. So BioPython lets us do kind of three major things. Lets you handle sequences of stuff, right? So in biology, you have DNA sequences and RNA sequences and protein sequences, and basically it has a bunch of useful functions that help you deal with all that stuff, all right? Another major sort of part of this, this package is dealing with 3D structures, okay? So molecules, right, exist as sequences of letters, right? A, C, T, G, T, G, A, whatever. But then when they actually behave inside your cells or whatever, they actually take on a shape in space, all right? And it's very important to know what that shape looks like. And there's a lot of data out there about different molecules' shapes, or also known as structures. So BioPython helps you out with that. And there's also population genetics, which basically, it's more of a mathematical thing, but it helps you understand how collections of organisms interact and how their sequences vary, or what kinds of patterns we can see arising from, I don't know, some sample of bacteria, right? It's going to be a population of sequences. So there's a bunch of things you can do in that area. I don't think I'm going to cover anything about population genetics. I'm sorry if anybody was interested in that. But there's a lot of math in there, and it would mean a lot of like background that we'd have to look at. So I think we're going to focus on handling sequences and 3D structures, and we'll leave the population genetics up to you guys if you're interested in it. So yeah. Here's some links to their very big BioPython tutorial, the BioPython website, and the wiki. So really good references. Most of my examples, I took them from there. All right. So the main sort of fundamental object that BioPython defines for you, right? It's just a type, is called a sequence, right? It's when you're dealing with sequence objects, okay? And basically, it's just a string. So you could forget about BioPython and do things directly with strings if you wanted, but often this can get cumbersome. And we can have 3 types of sequences, all right? We can have DNA sequences, RNA, and protein sequences, all right? And basically, they can all behave differently in the cell, so BioPython actually lets you treat them a little bit differently based on what type of sequence you're dealing with, all right? But let's say we wanted to create a very generic sequence. So we just say from bio.seq import seq, right? This is going to import the sequence class, or seq for short. And right away, we can start creating sequence objects, right? Kind of like what we're doing in the assignment, where we have an RNA object and it has a sequence. Basically the same thing, right? In retrospect, maybe I should have gotten you guys to use BioPython, but that's okay. Basically, okay, so this creates an object. It looks exactly like how you should have in your assignment, except for RNA. And the init function takes as input only, at least mandatory, only a string, okay? So I want to store this sequence, this particular nucleotide composition. I call the class seq, right? And it's going to create an object, okay? And if I printed the type of the object, right, I would get something like this. So it's a class bioseq.seq. And it has a bunch of attributes, okay? So this is the useful part of BioPython is that, for example, the sequences we defined in RNA, they had one attribute basically, which was their sequence or their structure. BioPython makes a bunch of useful attributes every time you create a sequence, and you can use them to do cool things. So what I did is I printed all the attributes of the object, right? If you remember the function dir, and then you give it an object, prints out all the attributes of an object. All right. So if you don't know what an object can do, you just print that, and basically it comes with a bunch of not very useful attributes. See all this underscore. All of these are kind of default. Every object usually has these. They're not super particular to our current object. So all I did is I just filtered them out and made a new list that has all the attributes without these underscores, and we'll get sort of what's actually interesting about the sequence, right? So this is just I'm taking the list of attributes and I'm appending it to a list, only if it doesn't start with an underscore, right? So here we are. So we have things like alphabet, back transcribe, complement, find, similar things like split, similar things to string. You can transcribe, translate, all right, upper, lower. So it behaves a little bit like string, but it has some extra functions that you might find useful, right? So right now, like I told you, there's different types of sequences, all right? BioPython doesn't know that this is a DNA sequence. It doesn't guess. It just says this is a generic sequence. So its alphabet attribute is set to just generic alphabet, okay? But if we know what type of sequence we're dealing with, it's useful to tell BioPython about it because we'll be able to do different things with it, okay? So let's just make 3 different sequences, all right? And basically, there's these objects called alphabet objects that BioPython uses to know which type of sequence you're storing, all right? You can just import these from bio.alphabet, and I'm importing DNA, protein, and RNA alphabets, okay? So sequences obviously are collections of characters from a certain alphabet, right? So English sequences have an alphabet X, A, B, C, D, E, F, G. DNA sequences have an alphabet of 4 letters, A, C, T, G, and so on. All right, so BioPython wants to know about that. So you can optionally pass one of these objects that describes what alphabet this sequence belongs to. Okay. So this is just the same as our previous example, except now we're telling BioPython this sequence is a DNA sequence, this sequence is an RNA sequence, and this sequence is a protein sequence. Okay? So let's just run that. Nothing should happen. It's just storing some objects. That's good. But now BioPython knows the difference between all of them. So obviously, DNA doesn't behave the same way as protein, and RNA doesn't behave the same way as DNA. So we can start doing biology stuff. Okay? So for example, if we have a DNA sequence, we can transcribe it, right? So what happens when you have a DNA, you have some sort of gene, and then that DNA gene gets converted into an RNA, which is almost a copy of that DNA sequence, right? And that process is called transcribing, okay? And basically that's a function that's implemented for you, where you can say, all right, I have this gene, right, which is just some BioPython sequence, and it's of type generic DNA, all right? So BioPython knows it's a DNA, so I can transcribe it. And I just say my gene.transcribe, okay? So this would be an instance method, right? Because it's acting on a specific instance of the class sequence. Yeah? Okay. So let's see if it actually transcribed this. So ACTA, what should be the difference, you bio people? How should the transcript look? This is not a computer science question at all. Yeah, exactly. Okay, so this is a straightforward one. RNA is the same as DNA, it's just the T's are U's, okay? So let's do that. All right, so first thing I printed was the transcript. So I say my gene.transcribe. It returns a new sequence object, but this sequence now has U's where we had T's, okay? So that's cool. If I give you a sequence now, you can transcribe it for me and tell me what the RNA would look like, all right? And if I wanted to get its alphabet attribute, so basically what type of sequence it is, it would tell me that it's from the RNA alphabet, okay? And let's say I have now that I've transcribed this gene, right, I have the RNA. The cell then from that gene is able to produce a protein, right? Protein has a different alphabet, but you're still able to go from a transcript into a protein. So you can do the same thing, right? If you have a transcript, you can say .translate, right? Again, that's an instance method because it's acting on an instance of the class sequence. .translate, okay? And let's say if I print it, right? Now I'm getting, if I print the resulting protein, I get this TSSG. Anybody know what these amino acids are?
Serine, glycine, and
What's the T?
yeah.
Threonine? Okay, nice. Thanks. Okay, so that's it. So it took your transcript and it did a fake translation of it, right? You know how translation works. You basically go codon by codon, 3 by 3, and that maps to a specific amino acid, right? So this would be a good exercise to do it in Python. I would recommend you do it like for practicing for the exam or something. Write your own translation and transcription functions. It'll just help you practice string manipulation and stuff like that. Okay, and now we know that it's from a different alphabet. It's from the protein alphabet, okay? We can also go straight from DNA to protein. We don't need to convert the DNA to RNA and then to protein. So we can say, all right, this is our DNA sequence, right? It's our gene. It's a generic DNA. And we want to get a protein from that, all right? We can translate that too. So we can just say my DNA.translate, and that should give me a protein. Okay, there we go. So methionine, alanine, isoleucine, valine, methionine. I don't know G. Which one? Glycine? Nice. Arginine? Lysine. Okay, nice. Yeah, should have known that. All right. Okay, so that's cool. We have a new sequence now, and it's a protein sequence based from this DNA sequence. And there's these little stars here. Anybody have a guess of what those stars are? So it's basically a stop codon, all right? What happened is, I don't know the actual codons, but there's a specific combination that tells the cell to stop translating that protein. And it doesn't really code for any amino acid, it's a stop. So it doesn't stop the translation. It still gives you the corresponding protein, but it says at these positions, I found some stop codons. Okay. All right. So this is not a very realistic way to translate a gene because, if you see a stop codon, you're supposed to stop and kind of not keep producing your protein, right? So we can do that by setting this keyword argument when we call translate, saying toStop equals true, all right? And that tells it if you see a stop codon, actually stop the process, okay? So if I did that, I would only get the first bit right before the stop codon, right? And then the rest of the DNA that I gave it won't get translated. So, slightly more realistic here. We can even go further and say, when you have a DNA sequence, there needs to be a start codon and a stop codon, right? And that tells the cell, where the gene starts and when it ends, all right? And that's by setting this keyword argument CDS to true. I think that stands for coding sequence. So saying, look, what I'm giving you, this coding DNA, treat it as though it were a real gene, a real coding sequence. And say, if you see a start, that's when you start translating. If you see a stop, that's when you stop, okay? So I ran it, and I got an error, Y. It says, okay, TG, key error, TG. So basically, it's crashing because it's not really a valid gene, right? There's no start, or it's finding some stop codons in the middle. Usually, the other time I ran it, it was a more extra in-frame stop codon. Yeah, there you go. So it found a stop codon in the middle, right, which we saw before. So that's an error. It doesn't know how to translate that. If I give it a more valid gene, so for example, ATG I think is a start codon and TAG is a stop codon, and there shouldn't be any codons in between, that should give me the correct protein from that, right? Okay. Any questions here? So just convenient little functions, right? If I give you a piece of DNA, you can tell me what the protein's going to look like. It's pretty cool. So another thing that comes up often is do you want to manipulate these sequences? So do some operations on them other than just getting transcripts or proteins from them. And they sort of behave like strings, these objects, these sequence objects, okay? So you can do things like stick two strings together. So if I say seq1 and seq2 are just two DNA sequences, I can stick them together and I get a new sequence object that has both stuck to each other, okay? I can also do indexing on the sequence objects. So I can say, for example, the first sequence object, I'll go all the way up to the second index, and I'll stick that onto the last index of sequence 2, and that'll create this other sequence, right? So sequence 1 was this. So I went, okay, I want to cut out everything until index 2, so 0, 1. And then I want to stick that onto sequence 2 at -1. So we're counting backwards here, T. So we should have AAT. Right? So this is just the same thing as strings. Same as strings is sequence objects are not mutable. So you can't go in and say this base at this position is now a different base. So if I went sequence 1 and I went to say at position 2 I want to put a G instead of an A, right? I would get an error saying sequence object does not support item assignment, okay? So they're immutable objects. Now, you don't always want these to be immutable. For example, if we had done the RNA assignment, we probably would have wanted to mutate, so make some changes in the sequence. So you can convert your current sequence object to a new type of object called a mutable sequence, okay? So it's kind of the same thing, it's just that you're allowed to make changes in the sequence of this object. So I can say, my mutable sequence is equal to my sequence 1, so my first sequence object, which was just this DNA, piece of DNA here. And I can say, use the method toMutable, converts it to a mutable type, right? Now if I print it out, now it's no longer of class sequence, but it's of class mutable sequence, okay? Almost the same, but now you're allowed to make changes to it. So now I can say, look, I want the first base to actually be a G now instead, right? So I didn't create a new object. I really just, inside that same object, I made a change, right? Kind of like how lists are mutable. Right? Another useful thing is, for example, searching. So if I give you a big long sequence and I say, find this pattern inside that sequence, you can say, all right, my sequence, I'll use its instance attribute, dot find, and I just give it a piece of a string, a sequence. And it'll go into this original sequence, and it'll scan until it finds that pattern. Okay? I can also say, how many times does this pattern occur in the sequence? There's the attribute dot count, right, which basically tells you how many times this little piece of DNA occurs in this bigger one. Right? So if I run this, it should tell me, all right, myseq.find told me that GAA, I could find it at index 3. 1, 0, 1, 2, 3, GAA. All right? It only finds one occurrence, so once it's found one, it stops. And the count, I say, look for this. How many times does this occur? 2 times. Let's see, we got GAA here, and we got GAA here. Right? So it seems to be doing what it's supposed to do. So the number of instant counts of a pattern is non-overlapping. So it really just, like it doesn't, like once it scanned this, for example, it won't like scan this. It really just goes by fixed chunks, okay? That's just a drawback of this, okay? And I would say, just a little hint for the final here, implementing these things on your own would be helpful. Like implementing this, for example, find this string inside another string. You know, things like that are probably going to be on the final, you know what I'm saying? Okay. So, now, let's get a little more involved here. That's just kind of the basic functionality of dealing with a sequence, a piece of DNA or RNA or protein. You can store it. I'll give you, there's going to be some more about it, but you know, let's do a little example here of some more real-life biology. So, I've only very rarely worked in a lab, but when I did, I had to do this. You know, let's say you take some sample from a cell or something, and you extract its DNA, and you put it in the DNA sequencing machine, and it tells you, you know, this is the sequence that you've extracted, right? And it'll look something like this, right? What you get back from the lab is your sequence that you sent me is this pattern, right? And let's say you don't know where that comes from, or you know, I just gave you a random sample, and you have to tell me like what organism does that belong to. If you asked a biologist what to do, they would tell you to blast it, okay? Which is the same thing as asking a computer scientist what something does, is to say Google it, all right? Has any of you used BLAST before? A little bit? Yeah? Okay. Yeah, so yeah, if you've done anything in bio, you've used it. It's really a straightforward thing. Basically, what it is an algorithm for searching. So it kind of does the same thing as this, actually. It does the same thing as this. So it takes a string, right? And it looks inside of a giant, huge database of sequences for places where that string matches, okay? So imagine that this thing was like a giant, you know, terabytes of DNA sequences, and I wanted to know where does the one that I found in the lab match up to this database, okay? And the nice thing about the database that we're searching in is that we know what organism every sequence belongs to and some information about it, right? So there's databases that contain tons of sequences, and you know, biologists have gone in and said this sequence belongs to monkeys and this sequence belongs to chimps, and you know, so if I come up with some random piece of DNA, I would say, all right, I'm going to look for this sequence in this database, and I should get an idea of where that sequence came from, all right? So yeah, it's really just an algorithm that, you know, it does string matching in a big database, okay? And BioPython lets you kind of do this programmatically. But if you wanted to, you would do something like this, you know, the rest of your bio friends are going to be doing this. They're going to go on the website, and they're going to say, okay, well, I have this sequence here that I found. I'm going to do these clicks, and then I'm going to paste it into the website, and then I'm going to hit enter, right? And then it's going to take that string and look where it occurs in this big database. It'll take a few seconds, and it should get back with some results. I really just punched my keyboard for that sequence, so I don't know if it means anything. Yeah, there we go. Okay. So it looks like there's some matches here. For example, I don't know, it looks like this sequence is found in some Amphimedon, Queenslandia, kinesin-like protein. Okay, so I have no idea what animal that is. Maybe some frog or something. But anyway, so these are all these organisms that have this a similar sequence to the one we found. So now maybe we have an idea of what our sequence is about, right? Anyway. So that's just a tool that comes up like really often in biowork, and BioPython has a nice interface for that. And it's called bio.blast is the module, and it has this class, or yeah, I guess it's a class, NCBI www. Okay, so that stands for National something something Institute www, so online, so it's on the internet, right? And that's where this, that's basically this thing that we clicked on here, right? It's just a database with a bunch of sequences, and you can send it a sequence and it tells you what matches it, okay? And we can do that really quickly by saying ncbiww.qblast, all right? You tell it what algorithm you want to use, and in particular this is called BLAST-N, N for nucleotides. So if you're searching DNA sequence or RNA sequence, it's an N. If it's a protein, I think it's a P. And this is the ID of the database that you want to search, NT. So all nucleotide sequences I want you to search. And then you just give it a sequence object, okay? Right, so we build our little sequence object. We tell it that it's a DNA object. And if we run that, it's going to do all the annoying clicking for us and come back with a result. So the result's going to come back, and it's going to be in an annoying format called XML. Basically, we don't have to worry about it. Okay, so we seem to have gotten a result. And basically, the result is kind of like a file that we receive, okay? And we can look at that file, but it's going to be messy. We don't want to look at it. Again, this bio.blast module has this object called NCBI XML. So it's basically for dealing with whatever the server sends us back is going to be in the format XML. And we want it to parse it. So parse means take a big file with some sort of format and extract the useful information from it. So all you have to do is say .parse, and we give it the result that we got back from querying BLAST, okay? Right, so that should have kind of gone through all the annoying bits of opening the file and looking for specific strings and whatnot. And now we have something called a BLAST record object, okay? Which basically is a nice container for the results of this search, okay? And like we saw, obviously, we got a bunch of results. Every one of these links is a result, okay? So each one of these is going to have its own BLAST record object with some attributes that we can conveniently access, okay? So for example, so we're going to go through every one of these records that we received, B, and then each one of these records has an attribute called alignments, which is going to actually show us like how the sequences match up. And then for each one of those alignments, we're going to print some information about it, okay? I'm not going to go into all these attributes. You can look at the documentation, but for example, you can have a title, you can have how long the alignment is. The e-value just tells you how likely it is to have seen that sequence. And we can actually print the sequence itself. So if we do this, yes, nice. So now we got back the information from doing BLAST, right? So this attribute here, sequence, right, it tells us some information about it, right, the organism and, you know, its location on the DNA, how long this chunk of DNA is, and sort of look at, like, this is the organism and this is our DNA sequence, so it looks like they match up pretty well. And you can keep doing this for all the other ones. So this is that Amphimedian Queenslandica thing. Can't resist, sorry. Sponge. All right. It's a sponge. Okay. So, quickly I'm going to introduce you to this other module in BioPython, which lets us add on some more useful information to these sequence objects. Okay, so right now all we stored about our sequences was really just the string of letters, and what type it was. But sometimes there's more information, right? For example, what species it's from, where is it found in the DNA, how it was extracted, whatever. All this additional information that can be attached to a sequence, right? So we want a convenient way to access that, and that's with this object called seq record, which is kind of just an extension of the just seq on its own, right? But it lets us put in some more information. And seq.io basically lets us sort of parse files and write files once we've dealt with the records, okay? So basically we want something to practice with, so I figured let's look at the bubonic plague. There's this database, again, of sequences from a bunch of different organisms. You can type in an organism and it'll give you some samples from that genome. So I went and looked for the Yersinia pestis, which is the scientific name for the bubonic plague, which I found out killed 25 million people in Europe. It's insane. And now we know what its sequence is, right? So we don't have to worry about it. So basically, a lot of these sequence records are kept in what's known as a Fasta file, which I think Chris kind of showed you what it looks like. But basically, it's a very standard format that stores sequences, basically with a header. So every sequence in a Fasta file has a header that starts with this arrow, right? And then all on that line, you have a bunch of information, right? So this is the Yersinia pestis, and it was obtained by whole genome shotgun sequencing, and this is its ID, or whatever. So when you download these files, they usually come in this format. Oh, and then what would follow is actually the sequence itself. I forgot to put that in. But basically, Chris showed you the annoying way to do it. This is the easy way to do it. You import seq.io from bio, and it already has a function implemented for parsing these files. Okay, so you can take a file from one of these websites. I stored it in this file called plague.fa, fa for FASTA. We can look at what it looks like. Right, so it's just one sequence here. It's a normal FASTA file, the header, and then the sequence, and then another related sequence, and then and it's DNA. Okay, so there's two records here. We don't need to do any for loops or anything. We can just say seq.io.parse, and I give it the name of my file, and I give it what format it's in. So it's in FASTA format. There's other formats you can deal with, but we're going to work with FASTA. What that does is it stores all the information in this records, in records, and basically each one of these sequences gets stored as a seq record object, okay, which is that thing I was telling you that is like a sequence, but just with some extra information attached, okay? So I went for every RN record, so this gives us essentially a list of every record it found in this FASTA. So this would be one record, right? And this would be another record, okay? And every one of them is of type seq record, and we can see what attributes are in there. So I did the same little trick of removing the non-interesting attributes. And for example, we have attributes, we have references, a description, some features, some ID, like the name of the sequence, the actual sequence. So more information about this sequence, right? And yeah. Where should you store it? Anywhere you want. If it's in the same folder as your notebook, you just have to use its name. If it's not, then you have to use some path, right? Right? And tell it where that path is stored, where that file is stored, and it'll look for it, okay? So I'll stop here for today. Are there any questions? All right, we'll keep going on Friday. Thank you.